Welcome back to another informative video on the Demystifying Research McMaster channel. Today we will be exploring the core principles of immunohistochemistry and tackle some common challenges researchers often encounter during its application. Immunohistochemistry is a laboratory method that uses specialized proteins, known as antibodies, to target specific antigens within cells, playing a key role in analyzing cellular function and foreign substance detection. The cells are prepared for this detailed analysis through a technique similar to the idea of museum artifact preservation, called formalin-fixed, paraffin-embedded tissue preparation. In immunohistochemistry, you start by heating tissue samples to make the internal components, known as antigens, more visible, a step known as sample preparation. Next, you introduce primary antibodies, special proteins designed to target and attach to specific cell components. This helps in identifying what's inside the cell. Then, secondary antibodies are added. These are another set of proteins that bind to the primary antibodies, enhancing the visibility of the cell components. Finally, the detection phase involves using special markers that allow you to see the results under a microscope. Next, we will touch upon the purpose and importance of immunohistochemistry. Primarily, it is crucial for disease diagnosis. Immunohistochemistry helps in identifying the distribution of various antigens in tissues, a key step in detecting diseases, including numerous types of cancer. Furthermore, it is used in developing treatment plans and determining disease prognosis. This technique allows for early detection of several neurodegenerative and neuromuscular disorders, such as muscular dystrophy, potentially reducing the severity of disease outcomes. In addition to disease diagnosis, immunohistochemistry can be purposed for tissue identification as well. This can be towards research and laboratory methods, or for more clinical applications such as detecting infections in living organisms. Using immunohistochemical procedures, infectious agents can be identified within tissues through using immunofluorescence assay. So now, we touched upon why immunohistochemistry is important and how it serves a purpose. But how is it applied? Well, it relies on antigen-antibody reactions, essentially a chemical reaction between two protein molecules. When targeting antigens within a tissue sample, immunohistochemistry utilizes antibodies as a start. The antigen itself acts as a marker. If the antibody within the tissue sample identifies the antigen, it will then be binded to it. With this binding process, the tissue sample will be stained a color, which provides lab technicians with results according to the color observed. With this observation, disease diagnosis can be put in place effectively. While we have explored how important immunohistochemistry is, it isn't without its challenges. Let's explore some of the potential pitfalls researchers may encounter during the process. One common issue is nonspecific staining. This happens when the antibodies used in the test bind to things in the tissue that they are not supposed to. This can lead to false positive results, making it seem like something is there when it's not. Imagine trying to find a specific protein in a crowded room. Sometimes, the wrong protein gets a spotlight. A few things can cause nonspecific staining in immunohistochemistry. One big reason is that the places where the antibodies shouldn't stick to aren't properly blocked. Usually, blocking agents such as serum proteins are used to cover up these spots and prevent nonspecific antibody binding. If this blocking isn't done well enough, the antibodies might attach to other things in the tissue, leading to nonspecific staining. Also, not using the right antibody concentration or leaving the tissue in contact with the antibody for too long can add to the problem. Using antibody concentrations that are too high or incubating tissues for too long increases the chance that it'll stick to things it shouldn't. So, getting the right balance of antibody concentration and time is important to avoid nonspecific staining. False negative results are another concern. False negatives can happen when the test fails to detect the presence of a target protein in the sample, even though it's actually there. This can happen due to many factors and can mess up the interpretation of experimental results. A false negative result in immunohistochemistry is akin to searching for a needle in a haystack but failing to find it despite its presence, leading to the mistaken belief that it's not even there. One common reason for false negatives is poor tissue fixation. If the fixing process doesn't work well, it can change the structure of the tissue, making it hard for the antibody to reach the target protein. This can happen due to both under and over fixation. If the target protein gets covered up or damaged during the fixation process, the antibody won't stick to it properly, leading to a false negative result. Another thing that can cause false negatives is when the antigen retrieval process isn't done right. Antigen retrieval methods are done to help the antibody bind better in immunohistochemistry. If the reagents used for the retrieval aren't handled properly, like having air bubbles or being expired, or not being stored right, it can mean that the target protein doesn't get enough exposure. This makes it harder for the antibody to bind to it, resulting in false negative staining. Also, picking the wrong antibodies can lead to false negative results in immunohistochemistry. Antibodies have different levels of specificity and how well they stick to a target protein. If an antibody can't effectively recognize the target protein, it won't show up in the test or cause weak staining, causing a false negative result. But fear not, there are ways to overcome these challenges. To ensure that the correct concentration of antibodies used, there are a few considerations. Two issues may arise: either the concentration of the primary antibody is too high, or not enough antibody is bound to the protein. In the first case, the antibody must be diluted to the optimal concentration. If not enough is bound, then a higher concentration must be added, or incubating the sample with the antibody for a longer period of time at 4 degrees Celsius. To ensure that the procedure is running as it should and that proper storage of the antibody occurred, it is important to implement controls. A positive control would involve taking a section of the tissue that you already know expresses a certain protein. A positive result will indicate that your system is valid. Negative controls can also be useful. Using a section of tissue you know would not express an antigen and getting a false positive result would indicate your system is invalid. When it comes to storage, the suggested thickness of tissues being stored is 4 micrometers. Also, storing tissues for too long can cause issues. A tissue stored for over 2 months results in the loss of certain proteins, such as P53. Water around the sample can lead to the loss of antigens. Therefore, it is essential to store slides in a way that protects them, such as using paraffin or vacuum storage. Additionally, ensure that all water is removed from the slide before storing it. Something else to take note of is proper tissue fixation. As mentioned before, if done incorrectly, it can change the structure of the tissue and cause difficulty with antibody binding. Generally, fixation for 24 hours at room temperature is suggested for most tissues. You must also choose the correct ratio of tissue to the fixative, as this can impact the extent of immunohistochemistry occurring. A recommended ratio is 1:1 through 1:20. To retrieve antigens, heat-induced epitope retrieval is the most common method used. Different equipment can be used, such as heating plates or microwave ovens. When using microwaves, excessive heat can destroy the antigenicity. Determining the appropriate retrieval for antigen and antibody is important as it varies across each one. Generally speaking, the microwave oven's temperature is 120 degrees Celsius at 750 to 800 watts for 10 minutes. For a heated plate, it would be 100 degrees Celsius for 10 minutes. Finally, blocking nonspecific binding must be stopped. Prior to incubation with the primary antibody, the sample is incubated with a blocking buffer for 30 minutes to overnight at 4 degrees Celsius. The ideal blocking buffer can be 5 to 10% normal serum from the source species for the secondary antibody, because it contains antibodies that bind to reactive sites to prevent the binding of secondary antibodies to those sites. The last consideration to take into account is consistency. Most of these measures all account for the validity of your experiment, but ensuring that you follow the same steps each time increases the reliability. By understanding and addressing these potential pitfalls, researchers can harness the power of immunohistochemistry to unravel these mysteries of biology and medicine with greater precision and confidence. Thanks for watching. We hope this video has shed light on the nuances of immunohistochemistry, empowering you to navigate its complexities with greater understanding and confidence. Stay tuned for more insights from the Demystifying Research McMaster channel.